Okay, everyone. So I would like to welcome everyone to the Theoretical and Computational Biophysics seminar this week. We have Professor Josh Vermaas from Michigan State University joining us as the speaker. Before I introduce the speaker, again, I remind everyone to please turn off your mic at the beginning of the seminar. I encourage you to turn on and keep your camera on during the talk because again, that becomes a little bit more personal interaction with the speaker. If you have really burning questions, feel free to interrupt during the talk and ask, or you can actually type your questions in the chat box and I can try to moderate this during the talk. Otherwise, at the end of the talk, we're going to have plenty of time to address questions. For those of you who don't know, at the end of the talk, we are going to have a career-oriented kind of panel discussion or chat, let's say, very relaxed, no agenda, no format at this point, in which actually a number of former group members of the ET group will be participating. So please stick around for that useful conversation, especially students who are in the job market or postdocs actually. So with that, so let me introduce our speaker. So it's my great pleasure to introduce Professor Josh Vermaas. Josh was actually a group member in our group. He received his bachelor's in science in physics, biochemistry, and computational math from Arizona State University in 2010. And then we were fortunate that he joined our biophysics program at UIUC. So he received his PhD in biophysics in 2016. After that, actually, he received the, let me actually talk first about his graduate work, because I think he's an excellent role model for our graduate students. So first of all, Josh has published 15 papers from his graduate work with me alone, and 3 more during his graduate work with national labs and other mentors and advisors that he worked with during the summers. He received an NSF fellowship and a computational science graduate fellowship from Department of Energy at the same time, and he had to relinquish one of them. So he went with the DOE fellowship that allowed him actually, or required him to do summer internships at national labs, at several national labs, and they all fell in love with him after he did his internship with them. So immediately after his graduation, he received a prestigious Director's Postdoctoral Fellowship from NREL, National Renewable Energy Lab in Colorado. And I think he was offered also another position from another national lab. And he went to Colorado. He stayed there for about 3 years before actually he joined Oak Ridge National Lab as a computational scientist in 2020, where he was in charge of NAMD performance and availability for users in relation to the InSight program that is a very famous DOE program. And since January, he just started as an assistant professor at Michigan State University in the Plant Research Laboratory and Biochemistry. So as I said, Josh has been extremely productive and a great, actually, citizen. So I'm very proud that I'm almost on half of his papers. So he has now 35 papers already at the beginning of his faculty career. And he was involved actually and interested in both science, not only with projects with me, but also projects from other labs, from his colleagues, from his other student kind of colleagues. And he was always looking forward to new science. And also actually on the development side, he was very active. He has a paper, for example, on topogromax, converting topology between charm and gromax formats. And he continues to actually be mindful of the importance of doing some development as a good computational scientist. So with that, so I think it was a pretty long, I can actually speak 15 more minutes about Josh easily, but I don't want to steal his time. So with that, I would like to thank Josh for being here with us and look forward to your seminar, Josh. Please.
Thank you, Ahmad, for the kind introduction. So today I'll be talking to you mostly about work that I've done as a postdoc at the National Renewable Energy Laboratory, while talking about exploring biological mechanisms and materials to enable the bioeconomy through computational physics. So first off, if you've looked at my CV recently, you might be surprised, wait, why isn't this a COVID-19 talk? So because of the ongoing pandemic, of course, everyone's research priorities shifted a lot. And so in working together with my colleagues when I was at Oak Ridge National Lab, we got on the cover of JCIM recently, as well as working together with JC Gumbart on studying MPRO within SARS-CoV-2. Most of what I was responsible for was enabling giga docking on Summit. So giga is 10 to the 9th. So we want to be able to dock 1 billion compounds in about a day. Now, if you're familiar with docking calculations in general, you know that a lot of these are CPU oriented and they're really slow. So for instance, AutoDock4 is a reputable docking program. There's recently been a GPU port that was done in OpenCL that's much faster. However, we're working on Summit, which is a Power 9 architecture plus GPUs, and OpenCL isn't supported there. So together with NVIDIA engineers, we were working to accelerate Autodock GPU so that we could run 1 billion docking calculations within a day. This is a great computer science story, but the biophysics is still playing out, so I'm not going to try getting ahead of ourselves here. But basically, through a lot of computer science development work, we can bring things that would take, like here we have a set of 10,000 compounds that we're docking. It goes from about 2,000 hours to under 6 on a single GPU. So there's a lot of acceleration that happens through code development that I was happy to be a part of at Oak Ridge. But what I'm going to talk to you today about mostly is what I see as sort of the great sustainability challenge going forward. So right now, the economy is oriented around photosynthesis that happened millions of years ago. So water and CO2 came together within plants and photosynthetic organisms. Through photosynthesis, gets put into the animal food chain and metabolism. And as those animals die off and are fossilized, you eventually turn them into coal, oil, and natural gas, which we turn into fuels, plastics, pharmaceuticals, detergents, carbon fiber, all the nice things that we like to use to keep our economy going. However, that creates CO2, and our carbon dioxide concentrations have been trending upwards since the beginning of the Industrial Revolution, which has environmental consequences going forward. So what I would like to be a part of is to move through a bioeconomy where the CO2 and water and photosynthesis happens today rather than millions of years ago. And rather than using non-renewable fossil fuels, we use plants and creating a closed carbon cycle where the CO2 that gets emitted is immediately used back up again in photosynthesis to still make the same things that we need to keep the economy going. This is something that's really interesting for Department of Energy use cases where we as the United States have 1 billion tons of biomass that we grow every year, and we need to do or make the most use out of it that we can. All right, so again, I realize who I'm talking to here. We need to spend a moment talking about plants. So plant cell interiors, they're mostly water, just like all eukaryotic cells. But what we're most interested in from a renewable material standpoint are the plant cell walls. Plant cell walls are primarily made up of 3 biopolymers. So you have cellulose, hemicellulose, and lignin. I'm going to be spending a lot of time talking about lignin, so I'm going to ask that you try and remember, oh, where did my pointer go? My pointer went somewhere. The H, G, and S lignin subunits, so the paracumaryl alcohol in the upper right, the coniferyl alcohol in the middle, and the synapyl alcohol in the far right. Those are the H, G, and S subunits that are catalytically glommed together to make a larger lignin polymer, which sort of links together the cellulose and hemicellulose fibers in plant cell walls. All right, now from a utilization standpoint, we know what to do with cellulose and hemicellulose. They're sugars, you feed them to bacteria, you make ethanol and methanol. That's kind of a solved problem. But the issue is we have no idea what to do with the lignin. And that's what we're trying to address. All right, so what is lignin? It is the second most abundant biopolymer on Earth. You only have more cellulose in the biosphere. It's an abundant byproduct of the pulp and paper industry, so we've had it for a long time. If you isolate it, it looks kind of like this brown powder, or sometimes it'll be black, depending on the composition. And it's really a useful material, or a potentially useful material for making bio-derived carbon fibers, or flame retardants, or feedstocks for aromatic small molecules. Now, I say potentially because so far the running joke in the lignin community is you can make everything from lignin except for money, because we haven't been able to figure out a way of making it economical to utilize lignin. Now, I'm coming at this from a molecular simulations perspective, and this is an animation from the project, one of the projects I was on as a graduate student at Oak Ridge National Lab, where we're looking at a large assembly of biomass. So each one of these red strands is a cellulose fiber, and then these blue globs are lignin monomers and polymers that have glommed together and are sort of connecting together the cellulose fibers. And then those green guys are cellulases, which we would ideally like to have breaking down the cellulose fibers. Now, molecular simulation is great for these kinds of things, because we can look in greater detail at specific properties that are really hard to get at experimentally. So one of the things, if we sort of look in the middle at the top, is we can directly compute contact surface areas between different components in our systems. So about a quarter of the cellulose surface is covered in lignin. And we can also calculate, well, how much of this is attracted to what specific face of the cellulose. Other things we can do are look at the time evolution of the interactions between components. And an animation of that is shown on the left, where we're going from like these red circles that represent the cellulose fibrils are being connected by the lignin polymer shown in blue and the cellulases to sort of form a big mesh network over the course of our simulation. And we can also look at things like, well, if I'm a cellulase, how big of a run can I have before I run into an obstacle? And shown in the lower right, each one of the black lines represents one chain of cellulose, and the blue or the green things on top of those black lines represent the lignin or cellulases that are blocking the path or the clear path along it. So we can measure these geometric things that would otherwise be really hard to obtain by experiment. Now, that was sort of one of the first projects I had moving into this biomaterial space. But what I would like to sort of bring your attention to are some of the lignin modeling challenges that lignin specifically has. So lignin polymerization is not controlled like a protein or DNA synthesis. So it happens through radical polymerization. So what that means is that we need some sort of method to simulate lignin synthesis. And together with a group at MIT, we developed a kinetic Monte Carlo approaches to sort of build up lignin polymers from individual monomers stochastically. And then once you have these polymers built, you need to translate these methods to traditional molecular dynamics. So you need things like a force field. So we need to parameterize lignin. And you also need to build three-dimensional structures to start your simulations with. So this kind of brings us to lignin structure. So lignin polymers are just linked together monomers. So starting from the base monomers at the top, together with all these different lignin linkages shown in the middle, that's how you build up a lignin polymer. And sometimes you can have different end caps that are modifying the lignin, or you can link them to carbohydrates. But those are the things that we need to be able to have within our simulation. Now, previous force fields only explicitly parameterized 3 basic monomers and the most common lignin linkage. So we needed to work on the rest of them when I started as a postdoc. Now, to do the parameterization, if you've been around the group long enough, you might recognize the paper on the left. We just followed the same charm parameterization workflow that was developed by FFDK, but we had to do it at a much larger scale because you have multiple different compounds that are that need to be optimized simultaneously with the same set of parameters. So we're building a whole force field. We're not just doing one molecule. So you do your same water interactions and dihedral scans, and you fit your poses against a potential energy surface. And again, if you start from CGNFF, there are things it does well and there are things it does poorly. But once you do the optimization through a conjugate gradient method, you can basically match your QM pretty well. So this is the way I like to think about it, is that this is machine learning, but we never called it machine learning because you're doing parameter optimization in sort of old school conjugate gradient style ways. All right, so that brings us to the fact that these new parameters better match experiment. So one of the things that we'd like to look at are the enthalpies of vaporization. That's the table up top. So the literature enthalpies of vaporization are closer in our newly optimized force field, shown in the far right, than what we started with CGNFF. And additionally, the crystal structure packing for small lignin-related molecules is reproduced. Now, we have this big table in the paper, but really what I'd like you to think about is looking at one of these crystals. So if you start from the optimized crystal of small molecules, they will form this kind of box. And if you simulate it with CGNFF, as you see in the far right, the box deforms. Whereas our optimized force field, yeah, there's some box deformation that happens because you're simulating this at non-zero temperature. But basically, it retains the same sort of structure that you see in the original crystal. All right, so that's our parameterization scheme set up. What we're interested in now is building lignin. So there is no database of 3D lignin coordinates to start from, like the PDB. So instead, what you need to do is first build up a topology and then turn that topology of how the lignin monomers are bonded into a 3-dimensional structure. So the Broadbelt group at Northwestern has created a program to develop generate lignin topology libraries based on experimental observations, and they do it like through a decision tree. So first you have to pick what monomer am I adding and what linkage should I have, and once I've built up this stochastic library of 100 monomers, do they satisfy certain properties that we want the population to have? From this library, or alternatively using this kinetic Monte Carlo approach, we can build 3D lignin models for lignin of varying sizes. Now, the way we do that, I'd like to spend a moment, and because I think it's a really cool animation, is to how we build up lignin monomers, or sorry, lignin polymers. So first, we generate all the monomers and we put them on top of each other. That was the first thing that this animation showed. And because they're linked together in the way that's specified by the PSF, we can do things like generate coordinates by superimposing dimers. So if you start from 2 monomers that are linked, you pick one of the dimeric structures that fits. So lignin's racemic, so you have to pick from among many different structures randomly, and then fix any atomic overlaps or ring piercings that result. So this actually, the method that we use to do this is based on a conversation I had with Noah back at BPS a few years ago, using MDFF type techniques as well as FEP type techniques to automatically relax away potential ring piercings you might see within the lignin structure, because we have a lot of aromatic rings. Then you'd solvate and simulate and go about your business analyzing the trajectories you get out. All right, so there are going to be 3 projects I'd like to show you over the course of the next couple of minutes. So one is to quantify lignin binding to cellulose. That's going to be one of the projects that recently got published. Lignin polymeric behavior in solution, as well as lignin permeability across cell membranes. So to sort of preface the lignin-cellulose binding discussion, within plant cells, we all know that lignin and cellulose are in the plant cell walls, that are outside the cells. Do they interact? Well, if you talk to the NMR people, they're really skeptical because NMR can't detect it. If you talk to the NMR folks, they're like, yeah, we see a bunch of peaks. We see that hemicellulose interacts with cellulose and lignin, but we do not see any peaks that correspond to lignin and cellulose interacting. However, we do know that cellulose degrading enzymes work faster when lignin is removed. And if you think back to the demonstration I showed you earlier, if you remove the lignin, you have a longer runtime along the cellulose surface until an enzyme has to pop off and get around the lignin obstruction. So simulation indicates that lignin binds to cellulose and probably does so in the hydrophobic phase. All right, now, if you've been thinking about, if you haven't thought about cellulose in a while, you may not realize what I mean when I say hydrophobic phase. Because of a cellulose crystal, if you look down along the long edge, there are 4 different faces that could be exposed along the cellulose crystal. So if you look at the yellow or purple lines, those represent hydrophilic faces of cellulose along 2 different side edges. Or if the cellulose is cut differently, you can have the blue lines shown at the bottom, which are also hydrophilic. And that's because you have hydroxyl groups that are pointing to the outside of the cellulose, and those can readily interact with water. However, there's also the 200 face, which is hydrophobic. And that's because none of the hydroxyl groups are pointed up in such a way that they could readily interact with water. All right, so if you want to go and measure how lignin interacts with cellulose, there are a couple of different ways you can do it, but we decided to attack this the dumbest way first. So we're going to construct some infinite sheets of cellulose with different faces exposed to solution. So either the 100 face or the 110 face, or the 1 - 10 face or the 010 face. So again, only the 100 face is hydrophobic, the others are hydrophilic. Place the lignin-derived compounds in solution. We're going to simulate for 400 nanoseconds and then determine the free energy or binding free energies of the compounds to individual faces. So again, because if you look at an individual trajectory, these lignin compounds will bind and unbind and bind again and unbind again, and they'll do this many, many times. We can calculate a binding free energy just based on how often is the molecule bound versus how often is it unbound. Because free energy is related to probability, we can just look at our trajectories and determine what these relative probabilities are. However, we have to make a choice about how we determine what is bound or unbound. So you can use something simple like distance and just say, okay, if the lignin center of mass is close to the cellulose center of mass, we're going to call this bound. If it's further away, we're going to call it unbound. So here on the diagram on the right, the bound section is in the middle, where either the cellulose midplane or where the cellulose midplane distance is small, or it's unbound if that cellulose midplane distance is large. We could also use the contact number, which measures the pairwise contacts between cellulose and lignin. So if the contact number is large, we say it's bound. If the contact number is small, we say it's unbound. The benefit to this is that it's not biased by lignin shape. So if you had a much larger lignin polymer than just the small little molecules that we were playing with or showing you before, like for a large polymer, the center of mass is distorted by what the shape of that polymer is. Because if you have a long skinny one, it may be contacting here at the bottom, but still have a center of mass that's really far up. But contact number avoids that bias. All right, so looking now again at our results for the cellulose binding for monomers, we look at the free energies of binding for all these different monomers that we tried. The 100 face tends to have the strongest interactions, which again, this jives with previous computational data that shows that lignin prefers the hydrophobic face. If you had to pick a hydrophilic face with which to bind to lignin, it would be the 110 face. What's interesting is ferrylate, which is the third from the right, which has 1 methoxy group, a hydroxyl group, and a carboxylic acid. That tends to like being in solution more than some of the others. And that actually fits in well with its physiological role. So ferrylate is the linker that's used to bind between lignin and hemicellulose. And because the hemicellulose is in the solvent, you need something that kind of sticks out in order to make that linkage happen, or allow that linkage to happen. So the other question is, okay, I kind of hinted at it earlier, how does binding scale with polymer size? So monomers and dimers are not good models for real lignin. Real lignin is polymers that are 10 mers to 50 mers to even sometimes 200 mers. So instead what we do is we actually draw from established lignin polymer libraries and then determine the lignin binding affinity. So if we have some 4 mers, we have some 41 mers, we have some 249 mers, and we need to figure out how well do they bind to a cellulose surface. Now, this brings up a technical challenge. Binding is spontaneous and irreversible on MD timescales for larger lignins. So I'm going to play this here in reverse in a minute. But basically, once something is bound, unless you pull it off, and in this case, we're going to do it through replicate exchange umbrella sampling on a contact number based reaction coordinate, you're never going to see this thing unbind. It will bind once and that's it, because the amount of time that we can sample is much shorter than the dwell time that you'd see typically. So we have to use a replicate exchange umbrella sampling calculations. And from that, we can calculate the free energies of binding. And we can plot it either against lignin molecular weight, as shown on the very far left. And that looks roughly linear. The R-squared values aren't the best. They seem to be better if we look at lignin-cellulose contacts. However, when I say lignin-cellulose contacts, what you should really be thinking of is contact area. So the contact number, even though it's defined through these pairwise interactions that are somewhat unintuitive, it correlates really well with the contact area between lignin and cellulose. So what this means is a lignin polymer that can pancake out and cover the cellulose surface is going to bind more strongly to the cellulose than one that remains compact as it interacts with the cellulose. So that's what I'm saying here. Pancake lignin would be the most stable. All right, so the key takeaways for this first part is that lignin prefers to bind to cellulose, the cellulose hydrophobic face. This might be hard for NMR to capture because the interactions aren't specific, and lignin becomes stickier as it makes more contacts with the cellulose surface. So altering solvent would change the structure and disrupt some contacts. This brings us then to the next question of like, okay, what happens if we simulate lignin polymers in solution? And again, the sort of methodology here is let's go start with some newly built isolated lignin polymers in solution. How does their behavior change depending on the molecular weight? Does the linkage structure or topology of the lignin matter? And what happens if you change the solvent environment? So first, it helps to kind of look at what happens to lignin in water. There are 2 basic dimensions that we can study here. One is looking at lignin molecular weight. So up at the top, I have 3 copies of a lignin polymer that are all about 2.5 kilodaltons. And at the bottom, you have 3 different lignins that are about 10 kilodaltons. The other dimension we can sample over are the topology of the lignin. So miscanthus is a type of grass, and its lignins are linear. Birch is a tree, and the lignin that it generates, it has a little bit of branching. Or you can have spruce, which makes its lignin highly branched. So we have these 2 different axes over which we can study. And well, we can look at a couple of different differences. So if we compare the mass versus the radius of gyration of these polymers, we see that there aren't particularly big differences. If we look at, they all are basically the same. The bigger polymers will change their shape more slowly than smaller polymers, and maybe more branching slows down shape changes as well. But there aren't some of the like wildly different things that you might expect overall. Now, in order to quantify how fast these shape changes happen, we're going to borrow some techniques from the protein folding literature. So we basically calculate a pairwise RMSD matrix and then cluster based on that pairwise RMSD matrix to figure out how fast the lignin polymers are changing their shape. So for the example pairwise RMSD matrix shown here, the cluster is identified by a color along the top of the pairwise RMSD matrix. And you'll see that it's basically block diagonal with a couple of reversions to previous clusters. Now, what we can do is taking that cluster banding, we can estimate how fast the lignin polymer changes its shape substantially. So that's shown here on the right. You'll notice that we have some trend lines, and you also notice there's a bunch of points that don't fit those trend lines. The reason we can't include the heaviest polymers is that there aren't enough transitions during our relatively short simulations in order to get enough transitions to be able to estimate a transition rate. But what we see is that between spruce, which is highly branched, and miscanthus, which isn't branched, they basically have the same mean state residence time for a given mass. So branching doesn't really change the relationship very strongly. Okay, now we can also simulate in solvents that are not water. Some solvents are known to be better than others for lignin, and commonly used pretreatments for biomass exploit this to remove lignin from other components. Now, how effective are known lignin solvents at changing polymer behavior? Well, we can kind of see this pictorially a little bit. I'd like to emphasize I'm showing the same polymer at the same zoom level, and water and hexane, water which is highly polar and hexane which is highly nonpolar, both are relatively compact compared with some of the intermediate polarity solvents you see in the middle. And if we go look at our polymer physics intuition now at different, for these different solvents, we see that the fitting exponent between the radius of gyration versus molecular weight depends on the solvent. So in panel B here, water has a relatively low radius of gyration versus molecular weight fitting exponent. Hexane has a similarly low one, and you get something higher in between. So in polymer physics, this means that higher fitting exponents mean that you have a better solvent environment to for the polymer that you're studying. All right, so the key takeaways from this part is that in isolation, lignin behaves like a typical polymer, and the standard polymer physics applies. Water will compact lignin into globules, and moderate polarity solvents are optimal for solvating lignin. But it doesn't particularly matter which of the nearly optimal solvents you choose. Now, this brings us to the third part for our lignin simulation overview, and that's looking at lignin permeability across cell membranes. Now, as I'm sure many of you are familiar with, is that active and passive transport are just really common across biology. And lignin synthesis is no exception. So we know how lignin is synthesized. It is starts from the phenylpropanoid pathway, shown in the right, and somehow the lignin precursors that are synthesized on the inside of the cell are assembled into a full polymer through lactases and peroxidases outside the cell. So somehow monomeric lignin has to cross the cell membrane. Similarly, if you're a bacteria trying to digest lignin, or a fungi trying to do the same thing, small lignin-related compounds that result from the breakdown of lignin need to cross into the cell. Now, some transporters have been identified to facilitate these processes, but not for all compounds. So it's an open question of how this is going to work. Now, the way that I'm going to attack this problem is to think about fluxes. So we're going to be comparing passive fluxes, the J passives, to the active fluxes, or the J actives. Now, to calculate a passive flux, you need the permeability coefficient, how big of a membrane surface area you have, and what the concentration difference is on either side of the membrane. For the active flux, that's basically a function of how fast the membrane transporters turn over and how many of them you have. Now, if we make some reasonable assumptions for transporter turnover rates and how many you can realistically support on a cell surface, you end up having this or creating a line. Now, if you have small concentration gradients and really small compound permeabilities, active transport is going to constitute the majority of the flux. If, however, you have a large concentration gradient or larger compound permeabilities, passive diffusion is going to be much, much faster. And through some estimates, you can say that along this black line, passive diffusion and active diffusion or active transport are going to be basically at the same rate, and you can go either way. And then this 10 to the 6th, the gray line that I'm showing in terms of compound permeability, that's going to be a useful demarcation later. All right, so we want to study the difference between active and passive transport. Attacking active transport directly to calculate permeability is not going to be feasible because we don't know what the transporters are and we don't know how fast they turn over. But we can calculate passive permeation through computational methods. And we can do this for 69 different lignin-related compounds, which are shown on the right. And we can do this for 3 different model membranes. So one is just a plain old POPC membrane, one is a membrane model for corn, and one is a membrane model for the bacteria Pseudomonas putida. All right, now we're going to back.
Could you, sorry to interrupt, could you remind us why you're interested in membrane transport of lignin as a polymer? So is that?
So it comes down not as a polymer, we're actually interested in the monomeric forms
because it forms outside the cell, I see.
Right, so polymerization in lignin happens outside the cell.
Gotcha.
The synthesis, because it comes from the phenylpropanoid pathway, which is how cells make phenylalanine. Somewhere in there, your monomers have to go from the inside to the outside in order for the biosynthesis to happen. So like that's the real driving question.
Gotcha.
We don't know how this happens. It could be active transport. There are some transporters identified, but is the question, or is the reason we haven't found other transporters because they don't exist or because we haven't looked hard enough?
I see. And then now that I interrupted you, how much do we know about the lipid composition of plant cells that you're gonna use?
Quite a bit. So like you have a lot of choices when it comes to your plant models. Pseudomonas putida was the bacterial model because that's what everyone at NREL was working on. I could have picked corn, I could have picked Arabidopsis, I could have picked many other things. The plasma compositions are kind of similar. One of the reasons I picked corn is because unlike it has a lot of sterols in it, whereas the bacterial membranes and POPC, they definitely don't.
Oh, okay.
So corn membranes have a lot of plant sterols, and that just makes it a different different calculation that you would, or you see different phenomena than you would in a model membrane.
Okay, thanks.
All right, so getting back to what we're actually going to be calculating, we're going to be calculating partition coefficients, which I'm going to be calling P, and that measures the distribution of products between membrane and solution. So this is often reported in the literature as log P. Now you'll have to remember that log P, when it's positive, means that you prefer to be in the membrane, and if log P is negative, you prefer to be in solution. I'm also going to be measuring the permeability coefficient PM. So that's a measure of how readily compounds cross the membrane. And the functional form of this depends on the free energy profile as the compound is crossing the membrane, as well as the diffusivity along that same reaction coordinate. And again, using replica exchange umbrella sampling, you can have 2 of your little lignin ninja stars moving up and down across through the membrane independently. And that's how we're going to be calculating the free energy profiles that we need in order to calculate both of these quantities. All right, so there's a lot of data that you can generate through this, because again, we have 3 different membranes and 69 different compounds. And what we do see is that the barrier properties will depend or will change depending on what the chemistry is of the molecules that we're interested in. So for charged species, in this case, I'm talking about ferrylate or coniferin, which are the purple or the red or purple lines in this graph. They show the highest barrier in the middle of the membrane. Whereas things that don't carry a charge, the barriers are substantially lower. So on the order of 5 or so kcals per mole, or in the single-digit number of kcals per mole. Diffusivity profiles, since that's the other component that we need to go measure, are pretty consistent, and mostly the differences are related to the molecular weight of the molecule. So coniferin is a lignin monomer with a glycosylation attached to it. So it's just physically larger, and that's why its diffusion is slower or measurably slower in our simulations. Now, the other kinds of questions that you can bring up is like, okay, if I look at a lot of these and I sample along one chemical group. So here I'm showing phenol, guaiacol, syringol, and catechol. So guaiacol adds a 1-methoxy group, syringol adds 2 methoxy groups, and catechol adds another hydroxyl. Why do they all have their free energy minima in basically the same place? And it basically comes down to hydrogen bonding. So the hydroxyl hydrogen bond, or sorry, the hydroxyl unit on the lignin likes to hydrogen bond either with carbonyl groups on the lipids or the head groups of the lipids themselves. And what you see is in solution, the individual monomers are basically oriented randomly. That's what a cosine of, or an average orientation with cosine theta being 0 means. But they will preferentially orient so that they can interact more with these potential hydrogen bonding donors as they move into the membrane. All right, so we can calculate a whole bunch of things and calculate the permeability and partitioning coefficients. However, big tables are really hard to look at, so I prefer to look at it this way. So as a scatter plot where the different chemistries are indicated by the color of the scatter plot dot. And what you see is the partition coefficients tend to be positive. So we tend to have compounds that like to be in the membrane. So if you're something that likes to digest lignin, you should totally be membrane associated. And if you're looking at lignin crossing, we see that most of the compounds, except for the charged compounds, have permeabilities that are greater than 10 to the 6 centimeters per second. So if we go back to the diagram I showed earlier, that means we are to the right of that gray line that I had showed previously, which means that if you if you're a bacteria trying to eat something at millimolar or higher concentration, odds are you do not need a transporter, because that's going to happen faster via passive diffusion than even the least permeable of the things we're considering. And if you're looking at something like benzene, where the compound permeability is around 10 to the 0, you have to go to very low concentrations before an active transporter can outpace passive diffusion. So this may be why there are relatively few lignin transport proteins identified, because the biology wouldn't waste its time developing a transport protein for something that it doesn't actually need to actively transport, or can't actively transport, because passive diffusion would outpace it. All right, so the key takeaways are that uncharged lignin-related compounds are quite lipophilic. So enzymes that act on these compounds may associate or interact with biological membranes, and lignin synthesis and degradation do not require membrane transporters. So organization of lignin synthesis by glycosylation may be one of the ways that how plants control their lignin topology by releasing a whole bunch of things at once, rather than having a steady drip of lignin compounds, because that has been shown by others to change the way that they assemble. All right, now the last 10 or so minutes, 5 to 10 minutes or so, I'd like to talk about a completely different topic that's beside lignin, and that's cellulases. And a really cool story I have of looking at the individual parts of the transfer of the life cycle of cell 7A. And because we were at NREL at the time, we had nice folks in the graphics department sort of show what the dinosaur of, or the dinosaur shaped enzyme of cell 7A looks like. So you have a head group with these yellow linkers that sort of move along as the dinosaur munches on the cellulose fiber below it. That cellulose fiber gets pulled into a tunnel-like cavity inside the enzyme, and then the dinosaur kind of poops out 2 units of cellulose at one time. All right. Folks had previously at NREL looked at different parts of this enzyme. So looking at its binding and association to the cellulose surface, looking at how it processively pulls the cellulose into the active site, as well as the catalysis of the active site. But one of the things that hasn't been addressed yet is how it dissociates from the cellulose, because again, if this enzyme runs into some obstruction, it has to dissociate and find a different place where it can bind to. There are 2 potential dissociation mechanisms you can kind of imagine. One is where the tunnel sort of slides off the cellulose, and we're going to call that the dethreading mechanism. And the other is if the tunnel sort of opens up, where the green and the orange residues of highlighted sort of separate and allow the cellulose fibril to fall out. And I'm going to call that the clamshell mechanism. What's nice in this particular instance is that dissociation rates for cellulose have been measured experimentally. And using simulation, we can compute the kinetics along both pathways and compare. What's even better is that the experimental assays measure vastly different rates depending on what assumptions they make and what techniques they're using. So we have a very wide target of potential K-offs that we could be measuring through our simulations. Now, the way that we're actually going to calculate what our K-offs are is to look back at a paper from the early 80s from Klaus and Zahn to figure out what the mean first passage time is for diffusive barrier crossing, which is what we're kind of looking at here. The way you do it is a double integral where you need to know the free energy profile again, W, as well as the diffusivity. So if you think back to the membrane permeability stuff we were doing previously, this is analogous to that, where you need to know the free energy, you need to know the diffusivity, and you need to take an integral. From that, you can calculate the mean first passage time is, and once you know the mean first passage time, that's just inversely related to the rate constants that experimentalists might measure. And like there's a lot of grunt work associated with coming up with this figure, but this is the key result. So since like looking at the free energy profile going from a bound state on the right to an unbound state on the left, we see that there's a much larger barrier for the clamshell mechanism that represents a state where the cellulase has opened and the cellulose is like halfway out, because you have to break a lot of hydrogen bonds simultaneously before water can really access the cellulose and make those or make more hydrogen bonds. So the barrier there is very high relative to what you'd see in a dethreading mechanism. As a result, for dethreading, your K-off is about 1 per second, which is in the ballpark of what AFM measured, whereas your dethreading rate for a clamshell type mechanism is ungodly slow. We're also very happy that our processivity estimate that we kind of get, so going forward from having a mostly bound cellulase to something that's catalytically ready, happens at the same rate as what we'd calculated previously by a different method. So again, what we see is that the clamshell barrier is significantly higher. And critically, we worked together with experimentalists to test some mutational evidence that also supports this hypothesis. So they prevented the clamshell mechanism by creating a disulfide double mutant, or that creates a disulfide linkage across the tunnel, preventing the tunnel from opening at all. And we see through mutation that the rate of dissociation doesn't change. All right, so with that, I'm nowhere near done with this. I'd like to go study much larger simulation systems and also use reactive MD techniques, including those developed through machine learning force fields, to break apart lignin and study biomass further. So molecular simulation will definitely continue to offer unique insight into these biological processes. With that, I'm going to open the floor for questions and thank the folks who helped me along this path. So Greg and Mike were my postdoctoral mentors, of course, Ahmad, a graduate student mentor, as well as Susan and Jeremy and Brandon, who worked with me at various stages in the national lab system. Questions?
Thank you very much, Josh. That was really fun. I guess I would like to comment that when you start thinking about these systems, very complex, structurally unknown, it looks like there is nothing one can do with molecular modeling and simulation with this. But I think it was very interesting to see how much you can still learn, or you have learned actually, by being insisting that yes, there is something we can learn, and how many papers you have published, how much insight you have got from these complex systems for which we have no structure. This is really amazing. You had to really use all tools of molecular modeling to bring pieces together and then do simulations to learn something about this. Is a problem actually that can also happen in other polymers, intrinsically disordered proteins and, fibril systems. There are many examples in which we really don't have accurate experimental structures to start with. And oftentimes we just give up and say, well, we can't study these things. But I think your talk was really a good example showing that you still can learn if you go about it sort of creatively and set things up correctly. So you can still learn about this system. Thank you very much. So I have one question, and then I can maybe actually invite people actually to. So I noticed your contact analysis. You had the power of 6 and power of 24 in one of your equations. What is happening there? So
okay, yeah. Hold on. Let me go back to that. Okay, so
there you go.
Yeah, so to be honest, that's mostly a function of like, well, you can pick whatever exponents you want there, right?
Sure,
I mean,
sure.
like a contact number is a made up kind of thing.
Of course, yes.
If you use like a standard, I think the default in Colvars is 612, or something like that, you end up getting a very steep drop off, and that's not necessarily what you want if you want to use it for biasing, because what you want is 0 to be very far away, but also that you're able to distinguish between changes in distance at low changes too. So you want something that's responsive over a long, or over a larger range.
So just make it less steep for your bias.
Yeah. So the 624, you could easily make that 626 or 622 or 824. It really doesn't matter. You just kind of
yeah, of course.
You try a bunch of them and you're like, this looks plausible.
Absolutely. It reminds me of Leonard Jones potential, which is whatever is easier computationally just put there essentially. Okay, thank you. Questions? So again, I think we're gonna keep it pretty informal, so you don't have to. You can actually just start. Everybody is now a panelist, which means you can turn on your microphone and camera and just ask your questions. So while people are thinking about the question, let me ask you another question. In terms of generating these structures, you said that you're using an algorithm that sort of essentially creates these branched polymers for lignin. So how many different structures would you get if you run this, I don't know, multiple times? I can imagine you really get an ensemble of different topologies, right?
I mean, so it's not so much the different topologies. So this part, like once you get to here, your topology is fixed, right? You have your topology from another source. It's either a lignin library where you have like 100 different monomers that you have topologically, or you've built it through some other mechanism.
I see.
But even here, there's still a lot of variability. So because lignin is racemic, every time there's a linkage, you have 4 different choices of geometries that you pick from, and I pick from them randomly because you don't know. So even from a given topology, you have 4 to the very large number different possible starting structures.
Yeah. I see. I see. I see. Okay. I see. Cool. Okay. Questions?
I have a quick question. It's Dhiman from UC Irvine. So it was a great talk, and thank you for this. It's a really interesting talk. So my question is, if we want to increase the timescales for the simulations that involves like multiple lignins and cellulose and multiple different things, and did you really consider like making a coarse grain model or somewhat like an implicit solvent model so that we can simulate a very long time scale process? And then like what are the informations that we might lose if we want to do that?
So implicit solvent you can do right now. The reason why we have stuck with explicit solvents is because one of the problems with implicit solvents is there's often nothing pushing them back in, right? And the small molecule interactions that you see actually matter for lignin. So like if we went to, where was it, this guy, right? So these nice compact water structures, I haven't tested it, but I suspect if you put this in implicit solvent water model, this lignin would just expand in a way that isn't quite right. Or alternatively, if it gets the water part right, it might be that it doesn't expand properly for methanol or ethanol. So these small scale stuff really matters. At the large scale, like that's kind of where we get to, where did I put it? This guy, right? So we have tried making coarse grain biomass models. As far as I'm aware, like there's nothing Martini-like. Like you could probably get away with some sort of Martini and analogous bead mapping to do lignin. But one of the things that experimentalists are really interested in is what's the difference between what's known as the S to G ratio. So what's the ratio of two different monomers? And the only way that those monomers differ is, oh, you added a methoxy group here. I don't trust Martini well enough yet to be able to distinguish those, right? It's certainly something that in the future there may be better methods of tackling this. But what we were interested in, like we could have simulated for a lot longer than 400 nanoseconds. Like the technology to do that is there. But what was more interesting for us was being able to simulate a large library of compounds and be able to say, we're not going to look at one lignin polymer because one lignin polymer isn't interesting. We want to look at a statistically meaningful ensemble of different lignin polymers. If that makes sense.
Yes, thank you.
Other questions? Stephen, yes.
I just have a question about, like, this is a very large polymer, right? In terms of the dimension of the system, how large is it? Just want to get an idea.
Let's see, the one that shows that probably most clearly. So the way I constructed my system, so this is like a 8 mer, and this is what 25 angstroms is. So like they vary in size tremendously. So on the big scale, for the largest polymers, those systems were on the order of 200,000 atoms. For these smallest ones, it was on the order of like 30,000. So there's a great diversity in how big your atomistic systems end up being.
And also, we build those highly branched ones because I would imagine this is is a high polymer system. It can easily get tangled. You have to be very careful in terms of because usually, because empty simulation is very slow, and usually, what you start with, usually what you end up with is not too much difference from what you started with. So do you have to be, very, very careful in terms of how you design the initial like whatever the branching conformation that you have for those lignin polymers?
So the only part where I'm careful is in making sure that we don't have ring piercings, because again, we were heartened to see that after like 20 nanoseconds, even if you start from extended polymer, in solution, it'll collapse to a globule. So you don't need a lot of time to get like ballpark reasonable structures. So, and like once you start from something that is globularized and you let it expand in a different solvent environment, that also didn't take very long. So in this case, I think we're kind of saved by the fact that a lot of lignin structures are plausible. Like it's not like an intrinsically disordered protein where you're like you have to wait for relatively slow bonds to rotate in order to really change your structure. All of our bonds that have to rotate are single bonds and they're fairly mobile. So that just makes the problem a lot easier.
Okay. And also I think I probably don't remember quite clearly, but at a certain point you said you tried a different solvent, right?
Yes.
Does this, in terms of this, I mean, how much does your result actually really, count on the sampling of this polymer configuration, I mean, conformation? Do you actually need to, I'm just wondering, like, in order to find easier ways to accelerate your sampling, like, did you actually try some kind of a mixed solvent or something similar to HMMM? I'm still having trouble with saying this.
So all of the solvents are real ones, because again, we're trying to, our goal was to have something that the experimentalists could understand. And like they know what DMSO is, they use it all the time. They know what THF is, they use it all the time. They used mixtures of like THF and water. I didn't show it here, but we have that data too. So like there were a lot of different directions where you could expand this further and say, we want to simulate these things for a microsecond each and like make that act like more than like 10 microseconds by using accelerated sampling techniques. But I am at the moment unconvinced that that would really change the result, because like the things that we were analyzing, or the things that are meaningful to analyze for these kinds of things, is like radius of gyration. Mm-hmm. That radius of gyration, if we simulate for longer, the error bar goes down. But ballpark, we have 100 of these different polymers that were, or 50 of these different polymers that we're analyzing. Even if the error bar for one of those goes down, the slope, or the estimation of the slope, won't change. And that estimation of the slope is what actually gives us the fitting exponent that tells us how good or bad we are. Okay. Or how good or bad that solvent environment is for the polymer.
I see. Okay, so whatever, because like the whatever the things that you're looking at is just not quite sort of independent of,
right. I mean, so like, unlike for like membrane transporters, where the time scale of conformational change is microseconds or longer, here, I mean, we have the data to say how long those conformational exchanges, or how fast those conformational exchanges happen, and it's on the order of tens of nanoseconds between things that are RMSD-wise very distinct.
Got it. Mm-hmm. Okay. Thanks, Josh.
All right. Any other questions about Josh's seminar?
Yeah, because otherwise we can get to the fun part.
Yeah, otherwise, if there are no more questions, thank you very much again, Josh. So we're